Okay, so this is for Anatomy 2, Chapter 24 of the Digestive System, and we're going to start Part 2, where essentially after the food has left the stomach, it is now going to enter the duodenum, the first part of the small intestine. And this is the intestinal phase of digestion. And the completion of digestion will be an effort between the contents that have just entered the small intestine, pancreatic juice released from the pancreas, bile produced and released by the gallbladder, produced by the liver, and all those things working together. So the pancreas, first of all, produces products that help in digestion. And digestion couldn't happen without the products of the pancreas and the products of the liver / held in the gallbladder, the bile. So the pancreas is located posterior to the stomach in the retroperitoneal space, so behind that peritoneum. It's oblong, it's elongated this way. And it's connected to the duodenum via something called a accessory duct or hepatopancreatic ampulla. Essentially, the production of, or the contents of the gallbladder / liver and the pancreas all join together to then empty their contents into the first part of the small intestine. The important things that the pancreas produces: enzymes that digest food, sodium bicarbonate, which will help lower the pH of the acidic chyme that's coming from the stomach. Remember, the stomach is lined with special or extra layers of mucous membranes that allow it to hold that acidic chyme. The duodenum does not have that same protective mechanism, so it produces the sodium bicarbonate that helps lower that, increase that pH. And here is just a picture of the pancreas, right here. And you can see here's the tail of it, the body, and the head. Pancreatic duct, so the products produced here will eventually drain into this duodenum right here, through this papilla. Then we can also see the cystic duct coming from the gallbladder and the hepatic duct coming from the liver. They join together into the common bile duct. Eventually both will join. The common bile duct and the pancreatic duct will empty into the small intestine. About 99% of the pancreatic acini, the glandular clusters, participate in exocrine secretion. So most of them are exocrine secreted enzymes, bicarbonate solution. The rest of it, 1%, form what are known as the pancreatic islets, the endocrine portion, so the hormone portion of the pancreas. So pancreas has dual roles in digestion and hormone secretion. And the hormones that the pancreas secretes are crucial. Glucagon, insulin, those have to do with blood sugar, somatostatin, and pancreatic polypeptide. We'll talk more about the endocrine system. So each day, about 1 to 1.5 liters of pancreatic juice are put into the duodenum. Like I said, it creates the proper pH in the small intestine. It also contains a starch digesting enzyme called pancreatic amylase that will continue to digest starch. Also, the following enzymes break down polypeptides, large proteins, into smaller dipeptides and the smallest building block amino acids. So those are trypsin, chymotrypsin, carboxypeptidase, and elastase. It also releases pancreatic lipase, which helps break down triglycerides. The liver is the second largest organ in your body, the largest gland secreting organ. 2 main lobes, the right and the left, a few smaller lobes in the back, and they're divided by that ligament that attaches it to the diaphragm, and it's covered by visceral peritoneum. It's made up of functional units called lobules. The hepatocytes are essentially the cells of the liver. You can see them here around. This looks like a star. They have digestive functions and non-digestive functions, just like the pancreas. So digestive functions include the synthesis, so they transform, and store proteins, carbohydrates, and lipids. They're also involved in detoxing of drugs, modifying excretion of exogenous and endogenous substances within the body or without. The bile that it produces also emulsifies, or that's what it says, breaks down the fats. The non-digestive functions, so those are the digestive functions, phagocytosis of old or worn out red blood cells. They actually make heparin, liver does, the anticoagulant, and other plasma proteins, fibrinogen and albumin, prothrombin, in the blood clotting cascade. When you cut yourself, those are involved. They're very crucial proteins involved in that cascade to clot your blood, and it also helps, along with the sunlight, to modify vitamin D to its active form. The liver is involved in that process. So what happens is blood is brought to the liver via the hepatic portal vein. Arterial blood is brought from the hepatic artery, and they come in to the liver here. So this one's on the way to the hepatic portal vein. This one is from the arteries here. And essentially the blood is filtered in through these sinusoids. It's a specialized capillary. So it's a pathway that the blood is routed through the liver. And you notice the hepatocytes, or the cells of the liver, are like the orangish cells, and they're on either side of this sinusoid. And they all eventually will dump into the central vein. Then we'll join the hepatic vein to go back to the heart. This just kind of shows the pathway of the blood through the liver and back towards the heart. Oxygenated blood from hepatic artery, including deoxygenated blood from the hepatic portal vein. They will both go through the liver sinusoids. Essentially, it's a filtering mechanism. Once it's filtered through these, it'll go through the central vein, then to the hepatic vein, up to the inferior vena cava, to the right atrium of the heart, and then thus re-entering circulation. So within those sinusoids, those specialized capillaries in the liver, you have these specialized, essentially immune cells. They're called Kupffer cells, and they just kind of are fixed there, meaning they don't move around the body. They are fixed in that location in the liver, a fixed macrophage, a Kupffer cell. And as the blood is filtered past them, they will help destroy red blood cells that need to be destroyed, white cells, or they catch bacteria that shouldn't be there, anything draining from the GI tract. The hepatocytes themselves around those areas are the ones that secrete the bile. And there you can see the arrow going this way is the bile secretion produced by these hepatocytes. And that bile will be released, and it will go out of the liver via the hepatic duct. And the purpose of the bile, like I said previously, is to emulsify fats, break them up, to get them ready for the watery environment. Or it helps emulsify them that are in the watery environment in the small intestine. So you need the bile to digest the fats further. Yeah. and hepatocytes produce about 1 liter of bile a day. Bile itself is alkaline, so not acidic, more basic. It consists of water, bile salts, cholesterol, and bile pigments. It's excretory product. It's a digestive secretion, both in the same. What the bile salts are used for is to emulsify the fats and also to absorb lipids. Without bile salts, most of the lipids will be passed out in your feces, so you wouldn't be able to digest. You would not be able to digest your fat that you consume without the bile salts. The pigment in bile is called bilirubin, and the color, it comes from the catabolism or the breakdown of old red blood cells. Like I said, here's the lobule, red lobule of the liver. The bile is being produced, and it goes into here's the right hepatic duct, the left. They join together for the common hepatic duct. And then eventually, they will come together to form with the gallbladder to form the common bile duct. What the gallbladder's purpose is, and it's on the underside of the liver, is essentially the liver produces 1 liter a day of bile. If you don't need to use it that fast, the gallbladder essentially is a storage device for the excess bile production. So it will hold the extra. It'll back up into there. And this is just a closer image of what we see there. The common bile duct is essentially gallbladder, liver, bile. Bile comes through there. And this is the pancreatic duct. And they will join together to form what's known as the hepatopancreatic ampulla, which will then, the contents of both of those, the bile and the pancreatic juice, will both empty into the duodenum at a location called the duodenal papillae in that small intestine. And this is about the backing up of the gallbladder in between meals, so when you don't need bile to be released because you don't have any fat going through your GI tract, the bile will back up into the gallbladder, and it gets concentrated actually tenfold in the gallbladder. And this just shows the pathway into the duodenum of those things that I talked about. So what causes the gallbladder to secrete bile? It's a hormone, CCK. Due to that hormone, that's what causes the release of that bile. Gallbladder isn't necessary for survival, but it is desirable. So you can live without your gallbladder if it needs to be taken out for some reason. You can live without it, but it is, you would like it if you could keep it. A colonoscopy, I can't say it right now. That's the procedure to remove the gallbladder. I know after somebody has their gallbladder removed, they need to adhere to a special low-fat diet and in smaller amounts afterwards because their body can't digest it properly right away. Okay, so we're in the small intestine now. We talked about the liver, production of bile, how that helps with fat digestion. We talked about pancreatic juice, the sodium bicarbonate, a few of the products there. Now we're in the duodenum. The first part of the small intestine is 10 inches long, not very long. The next part is the jejunum, 8 feet long, and the ileum is the 12-foot long region, the third region. This is interesting to note. The intestines are actually longer if you measure them in the cadaver, because in a live person, the smooth muscle is still contracting or capable of contracting, so it's going to be shorter. So there is a little bit of mechanical digestion in the small intestine. It's called segmentation. Essentially, the